Assalamu alaikum. Hello guys, welcome back to my channel. Today I'd like to explain about a parasite, a type of helminth or parasite we can say, that's transmitted to humans, from animals to humans even. And it's very dangerous. It's not that as dangerous as anthrax and brucella and other diseases, but it can cause death. So, the disease is Fasciola hepatica. What's Fasciola hepatica? Fasciola hepatica is also known as the common liver fluke. It's a fluke at the liver, or sheep liver fluke. It's transmitted to human throat. VIA means throat. First, eating raw meat or uncooked meat means when a meat is uncooked, the eggs may, or the cysts may enter your digestive system, GI or gastrointestinal tract. And second, plants and vegetables. Of course, contaminated plants and vegetables. So, we have a big life cycle of Fasciola hepatica. At your first year of college in veterinary medicine, even I think even in other medical colleges, at biology, at the first year we have biology, you will study this type of parasite, and in parasitology at the third year you will study it too. You will study the life cycle and you should memorize all. You will have an exam, of course, and at the exam all the pictures and life cycle of the parasites, morphological and microscopical examination are all included. But for the life cycle, the important thing is the 5 phases or 5 stages of Fasciola hepatica. The first one is egg, marasidium, and cercaria, and metacercaria, and at end the adult fluke. Diagnosis. How is this disease diagnosed? It's very easy to diagnose, of course. We will get a sample of feces or stool, we can say, a stool sample. It's already labeled when it comes to me. For example, I work in a lab, a microbiology lab, but we do some fecal flotation and other tests for determining blood parasites such as theileriosis and babiosis. We determine schistosoma. I will explain about schistosoma in other videos, of course. And schistosoma is one of the most important parasites in veterinary and humans too, and human medicine. The diagnosis: first, fecal flotation. It's a very easy test. I will put a video today about aseptic sampling of milk and fecal flotation, as I said in my past video. I hoped you to tell me the diagnosis of the video that I put it at the past. So, as we said, diagnosis of Fasciola hepatica or liver fluke is by first fecal flotation. It's very easy. By using saline, we bring a microscopic slide and a cover slip. It's all we need. There are 2 ways to do the test, but the most common way is using just a drop of saline, sterile saline, of course, and using a sterile stick, and putting a little bit of the stool sample. If you see, for example, some bloody substances, it's better to put that substance that looks abnormal. You put it on the slide, on the saline, and you make a suspension, just like a suspension, a little bit, and you will put the cover slip. I recommend to leave it for about 5 to 10 minutes to see the X more easier. And you can use, for microscopical examination, you can use oil immersion lens, or you can just use 40x lens, but the oil immersion is better, but even 40x can be used. It depends on the type of parasite. For example, in blood parasites, we should just use oil immersion lens. So we have fecal flotation. This is the result. We have egg of Fasciola hepatica. That's how it looks like. It has just one thin membrane in outer part, and it's oval shaped. And sorry, this is the adult form of Fasciola hepatica. It's easy to determine. It's known easily, Not, for example, coccidia. It's not just like coccidia. It's hard a little bit to find the species of Emeria and other species. This Fasciola hepatica is easy to find in fecal flotation. Even in veterinarian uses, we don't do some, for example, expensive tests, PCR and other serological. We don't. In our country, so we just do simple tests such as microscopical examination for Fasciola hepatica. This is the microscopical examination. Figure 2 shows egg of Fasciola hepatica, and figure 3 shows adult form of Fasciola hepatica. And a second way of diagnosing sorry guys, I'm a little bit sick, is post-mortem or PM. Okay, post-mortem is a common way of diagnosing diseases in poultry and even in ruminants, cows and sheep, camels. So this how it looks like. We can see the adult worms, as you see, guys. You can see it very clearly, and enlarged liver is seen. In the report, you can write enlarged liver plus adult worms of Fasciola hepatica is seen. You can write it in the report. So this is how it looks like, and it's very clear. We know that it's liver fluke. And one note, liver fluke may cause secondary bacterial infections, may cause other bacterial infections such as clostridial diseases in animals, other bacteria. The symptoms, 3 main symptoms. So, symptoms are loss of appetite. The animal will not eat. Of course, the liver is not working, so there will be problems at digestion. Enlarged liver, as you saw in the postmortem. Animal will not be able to stand, or paralysis, we can say. So in Fasciola hepatica, we have treatments. The treatments are tricalbendazole for cows and sheep only. Be careful. You can't use tricalbendazole for other animals. And albendazole, you can use it for mixed. Even albendazole sometimes may be used in pets in some cases. Actually, I'm not working with pets mostly. I'm working in a lab. I'm not working with pets actually. We work with mostly farm animals for diagnosing their disease. We have an important note. I write it by Arabic for peoples that are Arab and they don't understand English. The note is just as I said, tricalbendazole is just used for cows and sheep only. So, hope you get benefit. Thank you very much for watching my video about Fasciola hepatica, an important disease. Let's make a little bit of review. The treatments are tricalbendazole and albendazole. The diagnosis is by postmortem or fecal flotation if the animal is not dead, and it's transmitted from animals to human by eating raw meat and infected or contaminated vegetables and plants. So thank you.